So, good afternoon or good morning, everyone, depending on where you are. So, thank you so much for the introduction and the opportunity to present our work. Today, I'm going to talk about the expansion of chemical space and multiverse of natural products and food chemicals. This work is the team effort with the students of my group, in particular with the contributions of Alejandro Gomez and Karina Euridice. So this is the outline of the talk. First, I'm going to present a brief introduction on chemical space and chemical multiverse. I'm going to describe briefly this concept. Then in the second part, I will talk about natural products with emphasis in developments in Latin America. Third part, I will talk about food chemicals and epigenetic targets, in particular, the construction of a food chemical database. And at the end is presented a summary. So very briefly, we know that there are several definitions of chemical space. I particularly like in this one by Bishop and colleagues that state that the chemical space is a multidimensional Cartesian space, where the compounds are defined by physicochemical or chemoinformatic descriptors. In other words, we can understand chemical space as a table or as a matrix, where in the rows we will have the chemical structures, and in the columns we will have the descriptors. One of the implications of this definition is that the chemical space is not unique. It will strongly depend on the descriptors that we are using to represent our chemical compounds. Now, one of the most common applications of the chemical space concept is through the visualization. So if we again see the chemical space as a chemical space table, we usually visualize the chemical space as a, for instance, scatter plot, 2 dimensions or 3 dimensions, or as a networks. To do that, of course, we need to reduce the dimensions if we have more than 3 dimensions. Once we have the representation in network or 2 or 3 dimensions, we can also map a property in the chemical space, for instance, biological activity. And then, based on these visualizations, we can use them for several different purposes. Of course, that will depend on the goals of the project. Chemical space is usually used in diversity analysis of compound libraries to compare data sets in library design, and of course, to study structure property relationships. In particular, in drug discovery, is used a lot for the structure activity relationships. Now, in physics, the multiverse, as defined by Everett, is a hypothetical collection of different possible universes, each one defined by a set of specific properties. Now, if we put together the definitions in physics of multiverse with chemical space, we can suggest the concept of chemical multiverse as a group of numerical vectors that will describe the chemical space of a set of molecules in a different way. So in the slide is a schematic representation of the chemical multiverse concept. Say that we have a generic set of N compounds. Now, we know that we can represent those compounds using, for instance, drug-like descriptors. Or we can use a set of molecular fingerprints. And we know that there are many different types of fingerprints. Alternatively, we can use other type of descriptors, let's say constitutional descriptors. So if we do that for the same set of N compounds, we would have independent and, let's say, parallel chemical spaces, and each one will be a valid representation of this set of N compounds. So the chemical multiverse would be the group, the joint group of the parallel chemical spaces, each one defined by its own set of descriptors. For practical purposes, practical applications, the chemical multiverse will give us a more comprehensive or a more complete description of chemical datasets. So now I'm going to move to the second part. I'm talking about natural products, and then we will see how we are applying this concept of chemical space and chemical multiverse in this area. So we know that around the world, several different groups, companies have been developing natural product databases. Some of them are commercially available, but now in the recent years, many groups are developing public databases. And here in this section, I'd like to emphasize the efforts in Latin America. So Brazil, for several years, have been developing a very interesting collections of natural product databases. One example is the database of Nube that currently contains more than 2,000 compounds, and they will soon, the group in Sao Paulo State University and University of Sao Paulo is going to release a much more comprehensive database of natural products. Here in the slide, I'm showing part of the web interface of this database. In Mexico, for quite a few years, we have been developing another public database of natural products that is called DIFACQUIM. As in many other databases, the idea is to take advantage of the biodiversity, in this case of Mexico, the expertise of several groups that have been working in the isolation, characterization of the chemical structures. And here, basically, the idea is to put all the information together and make it public for everyone to see and check. The current version of DIFACQUIM has 553 compounds that come from plants, fungi, and marine animals. This database is available through the Zinc webpage. Here is a snapshot of how it looks like. Now, for the time's sake, I cannot describe each one of the databases of Latin America, but here in this slide, I'm showing a brief overview of the different databases that have been developing in the past few years. Here we again have Nube, DIFACQUIM. Another very important collection in Brazil is Systematex. Last year, Professor Scotti presented this initiative in this symposium. Also, El Salvador, Costa Rica, Panama, and Peru have been developing the databases. And now the idea is to join efforts and put together in a single database, in a single website, all these databases. This is how we are now developing in different groups a Latin American of natural product database, or in short, LANA PDB. The current version of LANA PDB contains 9 databases. Here is a list of the 9 databases from the different countries: Brazil, Costa Rica, Salvador, Panama, Peru, and Mexico. In total, LANA PDB contains nearly 13,000 chemicals. So we are analyzing the contents, diversity of these databases through an application of chemical space. So here in the slide, I'm showing visual representations of chemical space of LANA PDB based on properties. On the left-hand side, we have a visualization using principal component analysis. On the right-hand side, using Disney. LANA PDB is in purple. And for comparison, we are comparing Latin American natural product databases with Coconut. As we know, it's perhaps the largest public collection of natural products that is currently available. It is in orange. And also, as a reference, we are comparing Latin American natural products with a set of approved drugs. So visually, either in principal component analysis or Disney, we can see, well, as expected, that coconut covers a broad region of the chemical space. However, Lana PDB also has quite large diversity. In particular, this is more relevant in the Disney projection. So this is a visualization of chemical space based on fingerprints, in particular using max keys. Again, coconut as a reference, as a background, and also as expected, has a really large diversity, but Latin American natural products also contains quite few diversity. Also, we know that coconut contains around 400,000 compounds. Lana PDB, as I just said, has 13,000, and despite the fact the number of compounds in Lana PDB is significantly lower, it still has a broad diversity. This slide shows the visualization of the chemical multiverse of different and individual Latin American natural product databases. In this case, showing the multiverse as portrayed using max keys and map4 fingerprints. So each database from different countries are represented with a different color. On the left-hand side, we are comparing all databases in the single plot, and on the right-hand side, we are portraying individually each natural product database, different colors. The visualization of the chemical multiverse very rapidly is saying that databases from Brazil and Mexico are the most diverse. We know that currently these are the largest databases, in particular the databases from Brazil, but still the other databases from different countries are quite diverse. And I should mention that now we're actively working in collaboration with other teams to update and increase all databases. So this is the list of all the collaborators. So this is basically a joint effort. It's a multinational project. So of course, I'd like to give the credit to our colleagues in El Salvador, Panama, Colombia, Peru, Costa Rica, Brazil, and Argentina. So as we speak, the groups of Costa Rica and Colombia also have a major release of their own databases that will be implemented in the Latin American natural product database. Now, let me switch to the third part of the talk. And here I'm going to focus on chemical space of food chemicals and epigenetic targets. Now, we know that food chemicals have a really large overlap in the chemical space of natural products. And why epigenetics? Well, because also in my research group for quite a few years, we have been interested in analyzing the epigenetic activity of natural products and food chemicals. So the motivation of this project, it relies on the idea or on the field of epigenetics that we know is a study of heritable changes in the genome function without modification in the DNA sequence. Also, for the second time, very briefly, I'm just going to mention that there are three major epigenetic target families: writers, erasers, and readers. Now, a field that put together food chemicals and epigenetics is the field of nutri-epigenomics. That, as a definition, is a study of the interaction between food nutrients in the genome through epigenetic mechanisms. Also, we know from experience that regular food intake takes a major part in disease prevention, and we do not eat properly also can lead to several diseases. Now, the goal of this specific project is to put together a database that integrates the chemical information of food chemicals with epigenetic activity. This project is carried out using 5 major steps. First, basic literature search, but exhaustive literature search. Then the second major component is the construction of the database itself, and then the characterization of the contents, in particular, computing different continuous and descriptors and fingerprints of different design, analyze the content and diversity of the compounds in this library, and part of the analysis is the visualization of chemical space and chemical multiverse. Also, for the second time, in this presentation, I'm going to focus on the results on the first, second, and fifth sections. Briefly, this is a result of the literature search. So using standard literature search in Web of Science at PubMed, using the list of keywords presented here in the slide, in the period of 2017 to the current year, 2023, as of now. The results in Web of Science gave almost 6,000 publications. In PubMed, 730 publications. But using analyzing the, of course, we will have several duplicates here, but analyzing the contents, the results of each search, the results came up with almost 9,000 unique papers. So the second part was kind of very exhaustive work, extracting the chemical information, the epigenetic profile of all these papers. And just here, I'm going to present the results. The current version of the epigenetic food chemical database led us to select 184 unique compounds. So each compound in the database is annotated with the chemical name, structure representation using smiles, the food source of each component, the reference, and the epigenetic target profile. That is the key component of this database. So in total, the 184 compounds are annotated with 46 epigenetic targets, and this is a breakdown of each target family. So analyzing the frequency of the most reported targets, we cannot say that the most studied because perhaps not all epigenetic targets have been actually published, but at least what comes from the analysis of the literature says that DNMTs, DNA methyltransferases, that are epigenetic writers, currently are the most reported, in particular the enzyme DNMT1. Now, we know that the malfunction of DNMT1 is strongly associated with different cancers, in particular breast cancer and several others like cervical, prostate, colon, and lung cancer. And of course, here now the next step will, as I will mention later, will be study the structure epigenetic activity relationships using standard QSAR predictive models. Here is part of the analysis of the visual representation of the chemical space. This is using RDKey descriptors, and the visualization technique is Disney. The compounds from the epifood or food chemicals annotated with epigenetic target family are here in like magenta, and just for the sake of comparison, we are comparing the chemical space with the food database. That is perhaps the largest public database of food chemicals with overall 70,000 compounds. Now, despite the fact that the database of food chemicals with epigenetic activity is quite small, less than 200 compounds, Still, this visualization of chemical space very rapidly give us an idea that chemical structures are quite diverse, and we expect that as we expand the chemical database of food chemicals, the diversity will increase even more. So just to go to the discussion, I'm going to present here a brief summary. So in this talk, we mentioned the concept of chemical universe, of chemical space, that itself, we know it's a conceptual framework of cheminformatics with several practical applications. Chemical multiverse is the group of chemical spaces, each one defined by a given set of molecular descriptors, and the chemical multiverse is helpful to give us a more comprehensive view of the chemical space of datasets. Also discuss the Latin American Natural Product Database. Currently is comprised by 9 databases. The current version contains almost 30,000 compounds, and we have been inviting research groups from different Latin American countries. We know that there are several other countries that also have a rich biodiversity to contribute to this public and joint effort. And also, I talk about this epigenetic food annotated chemical database. It's a manually curated database with 184 food chemicals. All compounds are annotated with epigenetic target activity profile, and the immediate next step here in this project is to use this source of information to conduct structured epigenetic activity relationships. Now, the findings of both projects that I described briefly here have been posted a few weeks ago on ChemArchive. If anyone wants to check in more the details are publicly available, and now both works are under peer review. So with that, I would like to stop to go to the discussion. I'd like to thank all the students in my research group that is called DIFACQUIM, or Computer-Aided Drug Design at the School of Chemistry. In particular, I want to thank Alejandro Gomez and Eurídice Juarez, that also very kind, shared with me several of the slides that I show here. Also, other students actively participating on this project, Hassan, Fernanda, and Juan, and all of the group. And of course, many thanks to the funding agencies in Mexico and other countries. Finally, here is I'd like to share with you a picture of the main campus of UNAM in Mexico City, and this is the main library. Well, thanks again for your attention, and thanks again all the organizers for putting together this wonderful symposium.
Thank you very much for the nice talk and for perfect timing. Actually, we have a question in the chat from Vladimir Paroyko. Are all compounds in EpiFood database small molecules, or there are also protein, RNA, etc.?
Yeah, thank you for the question. The current version is only small molecules. But yeah, definitely, this is a good point. We can definitely expand with proteins or other biomolecules. Thanks for the suggestion, Dr. Vladimir.
Thank you very much. And actually, I have also a question.
Maria, there are 2 other questions in chat.
Oh, I'm sorry.
Just higher than the last one.
Okay. I'm sorry. Yeah. Are all 13,000 chemicals in LEN APDB unique, or there are some duplicates, etc.?
Yeah, these compounds are unique, and this was part of the curation. So it took, as you can imagine, several months to curate all the database, especially because we are retrieving all the information from the publications. And of course, by the way, we want to give credit to the original authors so that the database contains the reference to the original publication. But they are unique.
Okay, thank you. And also a question and a comment from our co-chair of this session. Very good work. How do you update the database periodically?
Excuse me, could you please repeat the question?
How do you update the database periodically? Well, it means that is this database, how to say, made and then it is already closed, or you somehow update it? Do you add some new compounds to the database?
Yes. So actually, this year we're releasing the first version with the contents I just described here. Now, in the second part of the year, again, as we speak, especially the groups of Costa Rica and Colombia have major updates, and we are expecting by next year, probably the first quarter, we're going to release the second version and also like put all the information in a public website.
Mm-hmm. Thank you. And do you have plans of extending this database to some other regions? I mean, not Latin American countries, but also some other.
Yes, so that's also a very good point. We recently talked with a group in Germany that have been developing Coconut, and since they have been assembling this very large database from different regions and different countries, we want to partner with them and work with them to put together Coconut and Lana PDB.
Mm-hmm. Thank you. And also there is one more question from our co-chair, and it's actually the same as I was thinking of. And how different are the phytochemicals of South America compared to other regions?
Yeah, well, we've seen that especially the natural products from Brazil, perhaps because it's the most heavily explored, has a unique compounds, kind of distinct compounds as compared to other countries in Latin America and also to other parts of the world. Yeah, especially from Brazil.
Mm-hmm. Thank you. And actually, another comment is more likely a suggestion of working together. Our co-chair, Sastri, he will tell that they developed OSADHI database with about 22,000 of phytochemicals, and they will be happy to work with you together.
Oh,
so it's a deal collaboration.
Wonderful. Good love to work with you. Yeah, definitely. Most happy to.
I will share the publications, and then it's very interesting that I'm looking forward to see how we can work together and then merge our databases and look into the similarities. And nice to see you and listen to your lecture.